In this video, we will discuss about how these intracellular galactins play a role towards triggering towards this apoptosis within the cells. So, from our previous videos, we have understood that there are two kind of apoptosis. First one is the intrinsic apoptosis, and another one is the extrinsic apoptosis. So, we will start with the intrinsic apoptosis. In this case, both the intracellular galactin 7 and galactin 3 might trigger the apoptosis, and how they will work in this case is that whenever the galactin 7 or galactin 3 uh, enters within the cytosol of the cells, it can interact with this BCL2 and form a complex. Galactin 7 interacts with the BCL2 with the help of this LND and TBIT, whereas galactin 3 can directly interacts with this BCL2 with the help of lactose. And interactions and the formation of this complex the will trigger the movement of cytosol or movement of cytochrome C, within the cytosols of the cells. And when the cytochrome C in the cytosols of the cells, it further down trigger the down regulation is further trigger the downstream signals because of the cytochrome C in the cytosol, and that will lead to the intrinsic apoptosis or intrinsic death of the intrinsic death of the cells. So this is how the intrinsic apoptosis works. In the case of extrinsic apoptosis, what happens when this first we will understand the function of this galactin A. The In contrast to the galactin 3 and galactin 7, galactin 8 prevent the apoptosis of the cells. So in this case, whenever this damaged vacuole or vesicles containing virus entered the cells or in the cytosol of the cells, galactin A interacts with this damaged SCV and helps in the repair of the damaged SCV, and simultaneously it forms a complex with NP52 and LC3 and interacts with the autophagy in such a way that the autophagy will cover up this damaged SCV, and thereby this autophagism will occur, and this damaged SCV will be back for the recycled back for the further regular protein formation. So this is how this galactin A prevent the apoptosis of the cells by autophagy lysosomes. In this case, the STC apoptosis will be triggered because of this irregulation in the endoplasmic reticulum, and we have covered this thing in many times, that because of the misfolded proteins accumulated in the endoplasmic reticulum, and this could be because of many reasons. One of the reasons that is mentioned here is because of the deficiency of GMAP2 or this alpha manosidase that detects the unfolded protein structures and divert them towards the ERAD, or proteins in this EDM family that will also helps in the degradation of the unfolded proteins from the endoplasmic reticulum towards autophagy or apoptosis. And dysregulations of these enzymes will trigger the increase in the ER stress because of the increase in the misfolded protein within the ER, and that will lead to, and one of the pathways that might get triggered because of the ER stress is the apoptotic pathway, and this apoptosis will lead to the cell death. And there is also there is also uh, there is also a idea that apoptosis might be triggered because of the accumulation of the unfolded proteins within this Golgi area. And some of the accumulations are mentioned here is about this Perlesian accumulation, and also this alpha manosidase, which does not have mannose 6 phosphate, and this is related to the ER. If it does not have mannose 6 phosphate, thereby they will not be able to filter the unfolded proteins towards the ERAD pathway, and that will lead to the ER stress, and ER stress beyond a threshold level will trigger apoptosis. And so this is how, when the mannose 6 phosphate is not working properly, then it might trigger autophagosome with the help of this LC3 2 enzyme, and that will lead to this polyubiquitinated protein into the lysosomes. And so this is in general idea. And this Becklin one is also leads to this autophagy lysosome. So what happens in this case is that whenever this perleation accumulation occurs, it will inhibit this Becklin one formation, and this Becklin one formation gets inhibited. So the unfolded proteins or the misfolded proteins will not be diverted to the autophagy lysosomes, and this will I mentioned it in the wrong way. So whenever this perleation accumulation occurs, that will inhibit this Becklin 1 formation, and that will leads to this polyubiquitinated protein formation. And this polyubiquitinated protein formation will be recognized by ubiquitination enzyme, and that will be diverted towards this autophagy lysosome. So this is because of this accumulation of perleation in the Golgi. However, these things are uh, not yet been accepted in the scientific community without any opposition. There is still doubts about this idea in the Golgi apparatus. However, other things are well uh, well noted and well documented in the literatures. So I hope you got an idea how this intracellular galactins and apoptosis function. And if you need to understand more about this intracellular galactins and apoptosis, you can go through the reference that has been provided in the description. You can also refer our previous videos where we discussed in brief how these galactins and apoptosis work within the cytosols of the cells. That's all for this video. Thank you for your time.